Thanks, Diane. This meeting's theme of vital connections in ecology is an excellent framework for us in these times of grand environmental challenges. Those of us who study ecology and work towards conservation often feel that we are walking a knife edge between hope and despair about our relationships with nature. If we lean too much on the side of despair, we risk immobilization. If we lean too far on the side of hope, we continue with the status quo. My understanding of vital connections started with my family connections. My father was from India, a Hindu, and a scientist. My mother was from Brooklyn, New York, an Orthodox Jew, and studied languages. My siblings and I embodied kind of a harmonious mixture of different religions and culture. And as a third child in this chaotic and large family, I found refuge in climbing trees. And I remember taking a solemn oath to learn about and protect trees when I became a grown-up. So I have always seen trees and forests as a sort of tapestry woven with the elements of other organisms and the environment into something that's like a real tapestry, something that's complex and connected, strong and fragile, something useful and beautiful. In graduate school, I chose to study the ecological interactions that occur in the forest canopy, which was then called the last biotic frontier. And today I'd like to share what my students and my colleagues and I have learned about the vital connections within forest canopies and how we have communicated about them to ecologists, to stakeholders, and to the public. I'll present this in three parts. First, the vital connections in forest canopies. Secondly, breaking connections through disturbance. And third, forging connections through public engagement. Most of this work has taken place in my two major field sites, the temperate rainforests of the Olympic National Park and the tropical montane cloud forests of Monteverde, Costa Rica. I'll start with describing the vital connections in the tapestry of the forest canopy. The microclimate aloft sustains greater extremes of temperature and relative humidity than the forest floor. The branches and trunks support diverse canopy-dwelling plants called the epiphytes, and also support canopy soils that are derived from epiphytes that die and decompose in place, which are classified as histosols. Compared to the forest floor soils, canopy soils are more acidic, they have lower rates of nitrification, they have similar C and N microbial biomass, and they sustain lower invertebrate density and diversity. I am most interested in the ecological and ecosystem functions that these canopy communities serve and weave into the forest ecosystem. In wet forests, large amounts of water and the accompanying nutrients that are naturally dissolved in that precipitation comes from allochthonous sources, that is from outside the ecosystem. And epiphytes have no root connections to the forest floor, nor to the host trees that support them. Rather, they've evolved mechanisms to intercept and retain nutrients dissolved in wet and dry deposition. They're absorptive leaf surfaces, as well as the tank form of many epiphytes, and the negatively charged surfaces of these arboreal histosols retain these nutrients for some time. After that, these allochthonous nutrients are then transferred to the forest floor via throughfall, via epiphyte litter fall, which is about 15% of the total litter fall, and by canopy roots. These adventitious roots emerge from branches and trunks of many species of host trees that efficiently conduct water and nutrients directly from canopy soils to their host trees. So overall, our understanding is that although the amount of epiphytes in canopy soils is relatively small compared to the whole ecosystem biomass, their vital connections allow them to participate in many aspects of whole forest ecosystem processes. Well, canopy communities also provide vital connections to arboreal animals. We monitored and compared the use of flowers and fruits and nesting materials between epiphytes and their host trees and found that 1/3 of all of their foraging visits were to epiphyte resources within the primary forest. And indeed, these vital connections of canopy entities with the whole forest have been documented mainly in intact primary forests. But as I'll talk about in this second part of my talk, I'll consider how forest canopy elements are affected by disturbances, both natural and human caused, at different spatial scales. Going back to the metaphor of tapestry, here are the threads of forest function on the horizontal threads. And we can also think about how potential disturbances may interact with those as vertical threads. I'll touch on studies of ecological succession at the branch level, community vitality and composition of microbial communities at the forest level, and finally bringing in other disciplinary approaches to understand disturbances at the landscape level. Well, let's start with responses of canopy communities to physical disturbances at the branch level. I carried out removal experiments on replicate branches and monitored the rate of return to epiphytes. Because they are so lush, I hypothesized that their recovery would happen rapidly. And every year I came back and measured percent cover on these recovering plots. Well, I was wrong about their resilience. After 22 years, only 40% of their cover had returned. And so although epiphytes appear to be robust, they're actually poorly adapted to the canopy environment. What about larger scale disturbances at the level of the forest? The canopy is a dynamic environment. Epiphytes fall sporadically to the forest floor as individual pieces or ride down broken branches and whole trees. For years, I casually observed that they died and decomposed rather quickly once they arrived at the forest floor. And I wondered whether that was the difference in the microclimate up there versus down below, or whether they're encountering different decomposer and pathogen populations that cannot disperse up into the forest canopy. So in 2012, I used an experimental approach I called the displaced limb experiment to understand the dynamics of fallen epiphytes in the temperate rainforest of the Olympic Rainforest Study Site. I hired an arborist to cut limbs into meter-long pieces as replicates for treatments. Those that my student are holding up are the severed segments that we used as our sample units. Here's our sampling scheme. Within the canopy, to account for the effect of attachment to the live tree, we compared responses of samples attached to live trees to samples on these severed branch pieces that we hung up in the canopy with strings. To learn the effect of being in the canopy versus on the forest floor, we compared canopy severed branch segments with replicate branch segments that we placed on the forest floor or on the ground. Over the 2-year study, we looked at a vitality index, which was based on color, apparent hydration, and the presence of dead material. We ranked these from 1 to 5 with calibrations from 2 different observers. We used a repeated measures mix model, which included time, tree, and experimental variables of attachment and level as the main effects. We found significant differences between the attached versus severed segments in the canopy for all time intervals. So connection with the living tree was important. And with one exception, which I point out with that black arrow, we also found significant differences in samples that were severed and hung in the canopy versus samples that were placed on the ground. So level was also significant. So what might be causing these patterns? Well, one obvious factor might be the microclimate. We do know that the canopy experiences brief dry down periods that do not occur on the forest floor. But differences in canopy forest floor microclimate would not explain the differences that we saw between tree attachment within the canopy. So what else might have caused these differences? Perhaps changes in the microbial populations of the soils that underlie these live epiphytes. My colleague, Billy Brazelton, is a soil microbial biologist. I worked with him and his student, Cody Dangerfield, to carry out standard 16S ribosomal RNA sequencing on the soils of our experimental branch segments. These are taxa-specific differential abundance plots or smear plots. They indicate the composition of different groups of microbe, and they differ between treatments. Each dot represents individual taxa. If a dot is black, it is not significantly different in abundance from the other treatment. If the dot is red, it is significantly more abundant in one population versus the other. The bars on the right represent broader categories of microbes like chloroplasts. The bars show us what portion of the red dots are in that category. So here is a graph of the canopy attached versus the canopy severed. Since there are lots of red dots, they are different in composition, and we can also see the different bar colors that their dominant group, but by the different bar colors indicate that their dominant groups are also different. Similarly, we found differences in the smear plots between the canopy severed and the ground samples. We also carried out whole community composition analysis. This multidimensional scaling plot allows us to identify broad patterns in the whole data set that might be correlated with our experimental treatments. The further the dots, the more dissimilar they are. So microbial community differences parallels that vitality index, those vitality index results, in that microbial diversity and composition of canopy soils were different from adjacent ground soils very soon after their transfer from the canopy to the forest floor. Therefore, the unique nature of canopy soil microbes is determined only in part by the physical environment. It is also determined by being connected to live branches. So these results represent an initial survey of microbial diversity of the canopy and provide a foundation upon which future studies can more fully investigate the ecological and evolutionary dynamics of these communities. Well, this study helped me realize that canopy communities can be venues to experimentally investigate disturbance ecology. The strategy of using model organisms in certain biological fields has advanced the understanding of complex systems. For example, Arabidopsis has helped us understand population genetics. Populus trichocarpa, black cottonwood, has helped us understand the genetics of drought resistance. And an analogous tactic in the field of ecology is to identify appropriate ecological arenas. The characteristics of these arenas are that, first of all, that they contain the elements of large-scale habitats, vascular and non-vascular plants, canopy soils, visits from vertebrates, arboreal invertebrates, and microbial communities. Secondly, they should be manipulatable and replicable within and between trees and forest sites. And third, they should have evolved under conditions that include multiple types of disturbances. Well, another type of disturbance that might affect the function of canopy elements in forests is forest fragmentation, which is caused by the conversion of intact forests to patchworks of forest fragments and pastures. This practice is rampant and increasing, especially in the tropics. I've been particularly interested in the extreme of forest fragmentation, where fragments become as small as a few or even single trees. Farmers do leave primary forest trees and pastures, what we call relic trees, for shade or aesthetics, for the fruit or medicines, or the wood is not good for lumber. So what are the ecological roles that the canopy biota might play in these altered landscapes? Do they retain those vital connections and ecological functions that we documented in the primary forest, interception of water and nutrients and providing resources for wildlife? Isolation of trees not only dries and heats the canopy environment, it may also isolate trees from vital connections to the primary forest in which they grew, from pollinators, from mycorrhizal fungi, and from fruit dispersers. Well, studies to date have produced conflicting answers depending on the site and the species. There's really no information for farmers and managers on how we might best configure these relic trees to maximize their potential ecological functions. We applied for a grant from the National Science Foundation, but it was declined because we weren't able to place these questions within the context of any generalizable ecological theory about the role of relic structures in the recovery of disturbed forests. So I thought that an interdisciplinary approach might help, perhaps fields well outside of ecology that are concerned with disturbance and recovery might have theory or practices or protocols or models that I could use. For example, the disturbance of a clear cut in a matrix of healthy forest might be analogous to the disturbance of a burn in the matrix of healthy skin. And so approaches to understand burn trauma and recovery of skin might help understand disturbance and recovery of forest fragments. So I recruited some faculty at the University of Utah from fields that deal with disturbance and recovery. We met for 2 hours a month for a year. During the first semester, each faculty gave a mini lecture on the approaches, the theories, and the tools that are used in his or her field. Then we spent the second semester articulating integrative and emerging themes. And I'll share just 3 of those today with you. First, for recovery of disturbed systems, it is important to extend and strengthen the web of relationships that concern it. Our faculty member in human development discussed this in terms of attachment theory. If an infant loses a parent due to death, that child often will have trouble forming trusting relationships as an adult. However, if the web of relationships of that family can provide an alternate figure, an uncle or a grandparent, a relic structure, so to speak, to take on the function of a parent, that can mitigate the negative effects of the disturbance of losing a parent. Well, this related to my ecological question about relic trees. My postdoc, Kimberly Sheldon, documented the number of bird visits to trees that are isolated in pastures in Monteverde. She found that the number of birds was directly related to the size of trees and their epiphyte loads. Visitation was also related to the distance to neighboring pasture trees. Visitation rates were not related to the distance to the forest edge. And so these results suggest that retaining large relic trees with large epiphyte loads and other relic trees in that habitat patch, which can foster the vital connections and the web of relationships, are important to visiting birds. A second insight from the colloquium was the importance of seeking knowledge from multiple sources. Our traffic engineering expert described how traffic engineers understand traffic jams, which are the disturbances of his ecosystems. They rely on many sources of information to predict and mitigate disturbances. Large traffic databases from Google Maps, traffic simulations, modeling, and interviews with drivers. And he noted that even though individual cars are discrete entities, when the cars move in traffic, they can be modeled by flows. And so to predict traffic flows, they rely on fluid dynamics theory, which is rooted in physics. And they have developed models to predict changes in traffic flow when a potential disturbance occurs, such as the installation of a new Walmart store in a neighborhood. So before a store is actually built, they can recommend where to place that installation to minimize traffic jams and to maximize flows. Well, we realized that we might be able to apply this to my relic tree question, how to predict the flows of birds when the position of relic trees are arrayed differently, clumped versus spread out. Can't do that experiment by transplanting trees. So we are now assembling a proposal for NSF across the biology and engineering directorates to see if this interdisciplinary transfer of theory and practice might be useful to ecologists. The third insight was about the consequences of disturbance. Most often, we tend to think of negative disturbances, the negative aspects of disturbances. We are all too familiar with the negative effects of the collective disturbance of COVID in terms of human health and the economy. But our colloquium group also recognized that positive consequences from disturbance can emerge. Our faculty member in refugee studies noted that even though displaced populations face many severe negative consequences, some are positive. For example, young women from South Sudan can get education in the new country of the United States, which might never have happened in their homeland. We realize that in some disciplines also, disturbance can result in unforeseen innovations. Our modern dance faculty stated that disturbance of one art form can be what she called a portal to the new. In dance, rejection and disturbance of the older traditional form of ballet allowed the new form, the innovation of modern dance, to emerge. And this led to the idea that systems almost never return to their original state. Our urban studies faculty provided examples of New Orleans after Hurricane Katrina. The image on the left is New Orleans in its original state, and then the storm created this disturbed, flooded state, number 2. And finally, with the advent of renovation and rebuilding, some neighborhoods prospered, and there was a decline in other neighborhoods in terms of infrastructure. But all over, we found that the disturbance created something else, the new New Orleans. Our colloquium discussions also help to see that humans tend to assume that a system, whether a forest ecosystem or an urban system or a human body, starts out in an original state, becomes disturbed from a fire or a hurricane or a car accident, and then eventually returns or recovers to that original state. However, systems often move to another recovery, to another disturbance, and then another recovery, and that it arrives at what we called a third state, or a fourth state, or a fifth state, neither the original nor the disturbed state, something different from both. And this is congruent with the ecological concept that systems often do not always refer nor recover to their original state, but rather are comprised of alternate stable states or alternate stable equilibria in ecological systems. Well, I think it's very exciting to think about new tools and new ways of thinking about disturbance, but it still brings me back to that knife edge of despair and hope. Why? Because at the same time that forests and trees are experiencing breaks with the vital connections of trees and primary forests, we are also seeing increasing breaks between humans and nature as we become more urbanized and more reliant on virtual rather than real interactions. Well, what does this mean to me as a scientist, as an ecologist, and as a person? I feel that I need to apply the precepts that I learned in the colloquium to mitigate the environmental challenges we are now experiencing. And so I'd like to share with you an approach that I've taken to extend my web of relationships, to seek knowledge from different ways of knowing, and to be open to a third state. And that leads me to the third part of my talk, which is about restoring connections through engagement. Well, public engagement, which is one activity for NSF's broader impacts or research impacts, is being promoted much more actively within academia, with high-level science officials and administrators calling for scientists to participate in public engagement. And for many years, I carried out public engagement in traditional ways with informal science education institutions such as the National Geographic, making television documentaries, creating a nonprofit called the International Canopy Network, and writing papers for natural history journals. And I think these are very important, but they tend to be viewed and read and taken in by people who already know the ecological values of trees and nature. And so I began recalling that my own value of trees arose when I climbed trees as a little girl. But I must recognize that a little kid growing up in New York City or in Tokyo may not have access to trees to climb. So what might she value that could be interwoven with trees and forests? Well, definitely Barbie. I actually myself was horrified when my 7-year-old daughter requested a Barbie, but her appeal is really universal. So in 2004, my students and I created Treetop Barbie, morphing this iconic plastic toy to embody the values of being a woman scientist who does research in the treetops. And in 2004, Mattel was not interested in that. So my lab students and I bought used Barbies. We dressed her in field clothes that were created by volunteer tailors. We created a booklet on canopy plants that accompanied her, and we distributed her on my tiny little academic website. But 15 years later, Mattel and National Geographic collaborated to create a new line of what is called Explorer Barbies. And I advised on this project, and they created a one-of-a-kind Nalini lookalike treetop Barbie. It sounds funny, but this experience led to the approach that I now use. People, I believe, will take care of and protect what they value, whether it's a Barbie doll or a sports car or an engagement ring or an old growth forest. So if we can articulate values of trees that resonate with people who might not hold ecological values of trees, we might be successful in gaining support for the protection of trees because of these other values that they hold. And my approach is to weave the values that I hold, the ecological values of trees and canopy organisms, with existing societal values that I might not hold. And this approach requires what philosophers call intellectual humility, the consideration of values that you might not yourself hold in order to find common ground with others. But I think that this goes beyond just the intellectual aspects of groups. So I call this humility for others' perspectives through engagement or hope. And what I'd like to do now is to describe the ways that we've tried to interweave the ecological values of nature and the scientific way of thinking, not by displacement or erasure of other values, but rather by recognizing that although our values might differ, respectful exchange can occur. And this brings me back to vital connections and this metaphor of tapestry. I can easily thread my loom with the many ecological values of trees and forests, the vertical threads. But I can also then interweave other existing societal values. We talked about interweaving the recreational and play values of Barbie with trees. And I've also carried out some engagement based on weaving spiritual values and the values of those who live and work in environments without access to nature. Well, what about religious values? Often science and religion are considered to be occupied two very different sectors and spaces, but 80% of the world's population self-identifies as believing in God or some kind of religion, much greater than those who self-identify as environmentalists. So finding common ground with faith-based groups about the values of trees could provide a way to at least to begin exchange. And although I was raised as a Hindu Jew, I am aware of the central image of the tree of life and the tree of knowledge in the Bible. And I use that as a starting point for hope, humility of others' perspectives through engagement with religious groups. I decided to use the venues and communication pathways and authorities of religious groups rather than those of academics as my authority. So I downloaded the holy scriptures of the world religions and did a search for references to the words tree and forest in them, since followers of that faith, well, they already have faith in those books. So for example, I found that in the Old Testament, there are 328 references to trees, and all of these verses related to values of life, analogies to God, ornamentation of temples, and practical use for food and shelter. And I found the same trends in other religions. For example, Jews celebrate Tu B'Shavat, which is the new year for the trees, with a ceremonial meal. Buddha found enlightenment under the Bo tree. Muslims seek out the form of the word Allah in the shapes of tree branches. And members of the Church of Jesus Christ of Latter-day Saints read about the tree with white fruits, which represent the love of God for his followers. Well, because I knew kind of next to nothing about what goes on in traditional places of worship, I spent several months just visiting them, sitting in the back pew and observing. And I saw that sermons were only 20 minutes long. They're delivered and received with emotion. People dress up for church and are attentive. Unitarians invited me in. They are delivered, let's see, there's music in churches. And at the coffee hour, very importantly, after the service, congregants were friendly and welcoming, even though I was not of their faith. With this information, I created a sermon on trees and spirituality and knocked on doors of churches and synagogues and requested that priests and rabbis allow me to give that sermon from their pulpits. I introduced myself as a scientist, but one who is interested in what they think and what they believe about trees. The Unitarians, as I said, invited me in first, and then other denominations followed. And in the 40 or so sermons that I've given, there has never been any conflict with creationism versus evolution, church versus science, because I drew upon their own holy scriptures as my authorities. Well, churchyards are considered sacred ground by their congregants. And so my students and I have mapped trees and churchyards and created booklets with both scientific information and scriptures about those species. I've expanded the reach of this by giving lectures in seminaries for clergy and training, which has led to tree plantings and bio-blitzes on seminary grounds. So the weaving together of ecological and religious values has been learningful and positive, leading to a third state between sectors that seem very far apart. Well, there's another sector that I have worked with, the world of corrections and incarceration, which really seem to be the antithesis of academia and the privileges that we enjoy. Over 2 million adults and 50,000 youth are now incarcerated in our country with very little access to science education. Prisons are also the most severely nature-deprived environments in which to live and work. And in 2004, I collaborated with state prison systems to bring science and nature to prisons in Washington State and Utah and across the country. We recruit scientists to give lectures on their research topics from astronomy to zoology. These monthly lectures are not for academic credit, but seem to provide strong educational values. Our evaluations on the effects of lectures on the incarcerated are based on over 2,000 responses to our pre and post-lecture surveys. These have shown significant increases in science knowledge content, shifts in self-identity towards being more capable science learners, and a stated desire to find out more about the topics that we present. We've also developed collaborations with conservation groups to include people who are incarcerated as citizen scientists, in which they contribute to rearing endangered species for existing restoration projects. All of these efforts are voluntary. Our first project involved the Washington Department of Fish and Wildlife, in which the incarcerated men raised at Cedar Creek Correction Center raised the Oregon spotted frog from egg to tadpole to adult with an 85% survival rate, which was higher than any other captive rearing facility in the state. Other collaborations have been with the Nature Conservancy to grow rare prairie plants, Hawkwatch to build nesting boxes for the American kestrel, and involving women who are incarcerated to collaborate with U.S. Fish and Wildlife scientists to rear the federally listed tailored checker spot butterfly. But the benefits go both ways. Our formal evaluations have documented that participating scientists have become better communicators, better teachers for those with diverse learning backgrounds. Many have stated that they have become more aware of social injustice and mass incarceration, and many have taken actions such as joining Black Lives Matter and writing to policymakers to increase funding for correctional education. Well, in this work, we have seen some small part of the despair that is ever present in the system of mass incarceration. We do understand that our activities and programs do not solve the persistent injustices that continue to exist in the world of mass incarceration, but providing access to science and conservation projects inside these institutions are actions that we as ecologists can provide. Well, when engaging with the public, there is this tendency to think about the size of the group and the level of impact that we are having. Many ecologists think that they have to create large projects to succeed in public engagement, but I think that that is not so. Small actions by many of us can have an impact. And in doing the math, where we know that there are 325 million people in the US, there are 6.2 million scientists. So when you do the math, if each scientist talks to just 52 people a year, that's just one person a week, we will have reached every single person in the United States. And here's one example of a small action. I noticed that many people pay attention to decorating their fingernails. And even though it doesn't really appeal much to me, I decided to go to a nail parlor. I asked the manicurist to paint trees on my nails with bright green paint. And then when people notice them, I could have a great conversation about trees and forests. So you might think about doing the same thing with your study organism and your fingernails. In 2016, my colleagues and I developed a training program for scientists called the STEM Ambassador Program, which is designed for scientists to carry out public engagement events in the venues of public groups, where they live, where they work, where they recreate, and where they worship. We help scientists identify particular focal groups that they might have common ground with based on their research interests, but also their personal characteristics and their experiences. So I've talked about the critical connections and canopies, about connections between academic disciplines and across societal sectors. I'd like to finish with a few thoughts about how the ecological disturbances of our planet and the social disturbances of our society affect the academic ecosystems in which scientists work. Academia is a long-standing area of privilege. We are now hearing calls to make the scientific enterprise more equitable, to broaden participation in it, to make it a more welcoming place for a greater diversity of people, both in terms of ethnic groups that have been traditionally underrepresented and in terms of becoming more interdisciplinary in thinking. In summary, to create and reinforce vital connections between ecology and society. I think we must foster an academic culture that manifests a third state in which we forge stronger webs of relationships and seek information from more diverse people and sources. It will take a long time and require work from many quarters, from university presidents to undergraduates, from funding agencies and from policymakers. What can a single scientist do, a faculty member like me or a graduate student like some of you? Well, let's think about the trajectories of a career in ecology. A trajectory means a path, a progression, or a line of development that moves you from one place to another. Points along that way can boost the speed and the upward direction of that trajectory. And we can ask what points along that trajectory will boost our efforts. Well, we already provide our graduate students and our emerging scientists by helping them map out a research trajectory. What papers should they read? What papers should they write? What support mechanisms do they need? What grants should they apply for? What contacts can you introduce them to? I believe that we should have these discussions, not only concerning their research trajectory, but also their research impacts trajectory. What public groups do they have an existing affinity with? What programs on campus might help them forge positive relationships with the public? Who in the University Development Office has contacts with foundations that might fund their efforts? Can you work with a department chair to get funding to bring a public engagement of science training to your department? Well, as a faculty member, you might not have these answers, in which case you could lobby to get a broader impacts unit in your chancellor's office, as many universities have now. You can encourage your students and your faculty that these activities are part of what a scientist does, not a burden and possibly a benefit. That a piece of research is not complete unless it is not only published in the scientific literature, but has some outlet to society. My colleagues and I are now writing a paper about research impact trajectories, and I invite you to send me information about what helped to boost your research impacts trajectory. You can send that information either to my Twitter handle, or you can write me an email, and I've listed those both below. Let me know what helped or what you think might help in the future to bridge ecology and society. In summary then, by weaving these individual threads of our efforts, we ourselves become forgers of vital connections in ecology and in society. Making and maintaining these efforts with those whom we work and converse and interact can together create a tapestry of humans and nature that is like a forest, that is like a real tapestry, something that is complex, that's connected, that's strong, that's fragile, that's useful and beautiful. Thank you.